everybody. I'll be talking today about relationship between two characteristics of a gene. One is its expression level, and the second is its compactness. So let me start by defining the two terms. So intuitively, a gene is compact if it's short. But a typical eukaryotic gene is made of different components, as you can see here, from exons, which are described by these blue boxes, and intervening introns, and on the two sides, the 5' and thus. So what exactly is short when we talk about gene compactness? So what I did is not to work with a single measure of gene compactness, but rather with a collection of variables to measure compactness. So one measure of compactness is the total transcript length, just the length of the entire transcribed unit from the beginning to the end. Another measure is the length of the coding part, the CDS length. It's just the length of the coding part of the gene. A third measure is the total exon length, including both the coding parts and the UTR parts. I have been also looking at total intron length and the length of two UTRs. So to these variables, I call the total length variables because they measure total length of different components of a gene. In addition to this, we have been looking also at median or average length of exons, of introns, and also about the intron density per length of coding sequence, the intron density in the gene. So the collection of all these variables is measuring how compact is the gene, and I will refer to all these variables along the talk. Now, you all have a pretty good notion of what is expression level, but what I wanted to do for this study was to characterize each gene by a single number, a measure or average or whatever you want to name it, expression level relative to the other genes in the organism. So to achieve it, we followed a procedure that is custom in this field. So we start with a matrix of expression values, and this matrix is per one organism, and we have the different genes along the rows and different conditions or tissues along the columns, and the numbers are indicating the measured expression level. And what we do, we rank the genes in each tissue separately. So just for this toy example, we pick 3 categories. C is 3. As you are going to see later, we actually work with more like 30 categories. But we pick a number of categories, and then we just rank the genes in each tissue separately. So in this example, the 1/3 of the genes that are most highly expressed, we give them the category number 3, and the 1/3 of genes that are lowly expressed, we give them category 1, and all the genes in between, we give them category 2, and we do it separately for each tissue. And then we just compute for each gene the average ranking. And the last step is just to round the number in order to assign an integer category for each gene. So this is the measure that we used to rank the expression level of a gene relative to the other genes in the same organism. So now we have a collection of varying compactness and a category level measuring the expression level. And we want to look at the relationship between these two characteristics. And the first work to do it was published in Nature Genetics in 2002. And in this work, they looked at human and C. elegans, and they looked only at intron length versus expression level. And they noticed that there is this trend. So highly expressed genes have shorter introns. And we will call this the C up E trend. For compactness goes up with expression level. The more highly expressed is the gene, the more compact it is. And they suggested in this paper an explanation for this trend. And they say that transcribing takes energy and takes time. So it's about 20 nucleotides per second, and at least 2 ATP molecules per nucleotide. And so those genes that need to be transcribed a lot, just because of selection, you have a selection pressure to find in them short introns to save time, to save energy. And this the explanation is called the selection hypothesis. And following this work came many other works looking at exactly the same relationship, and they all reconfirmed and validated this trend. So other people have been looking at mouse, at Drosophila in plants, and they were measuring not only intron length, but also length of exons in the gene, the length of the coding region, the CDS length, and people have been looking at length of the untranslated regions, and expression level in many different ways, by using ESTs, by using SAGE, using microarrays, and in all these cases, they reconfirmed this trend that highly expressed genes tend to be more compact. And the only challenge to the original work was the explanation itself. When Vinogradov looked at the same thing, he noticed something that could not be well explained by the selection hypothesis. What he saw that when the genes are highly expressed, the intergenic length gets shorter. And this is another way to say the now well-known fact that highly expressed genes tend to cluster together in certain genomic locations. So the intergenic region is becoming shorter, and this cannot be explained by the selection hypothesis, and therefore Vinogradov suggested an alternative explanation to the same trend. He said that we should look, instead of the expression level of the genes, we should look at the expression breadth. So in how many tissues the gene is present, is expressed. And he claimed that those genes that are narrowly expressed, that is, that they are expressed only in a small number of tissues, they require more complex regulation, because you have to regulate exactly in which tissues or which conditions they should appear. And therefore, those genes that are broadly expressed are more compact because they need less room to host regulatory elements. And it is well known that there is a strong positive relation between the breadth of the expression and the level of the expression. So because that broadly compact, you also observe that highly expressed genes are more compact. And to this explanation, he calls the genomic design hypothesis. And basically, he says that you are gonna see the same trend, but the driving force, the determinant factor, is expression breadth rather than expression level. The challenge to the trend itself came in a Trends in Genetics paper, where the authors looked at plants, at Arabidopsis and rice, and they reported exactly the opposite trend. They said that genes that are highly expressed tend to be less compact. And it was, they could not suggest any good explanation for this. They just say that maybe some other factors, some other forces are acting in plants, but this was the report. To summarize how things look so far, so we see that in many cases this C-up E trend was confirmed, that compactness goes up with expression, and there were two alternative explanations suggested to explain this trend. The first trend was reported in plants, but there was no good explanation for it. And I believe the outline of the slide hints that I'm going to suggest something else that is going on. And this is that I believe this to be the actual relationship between expression level and compactness. So you have indeed that highly expressed genes in this part are more compact, but so are genes with very low expression, and there is some bend point in the middle. And I believe that also some modifications of the original explanations can be used to explain this non-monotonic trend. So first, let me try to convince you that this is indeed the case also in plants and in animals. So what you see here is this. In the columns, you see different organisms. This is human and Drosophila and C. elegans and Arabidopsis. And the rows are 4 different variables that measure compactness: the total transcript length, the length of the coding part, the total exon length, and the total intron length. And you see that everywhere you see such non-monotonic trend. And in order to confirm this non-monotonic trend, we use a technique that is called the segmented regression, where we do linear regression but allow the data to be potentially segmented, and the linear line that fits the data can slope at certain points. And in all these cases, in all 16 cases that you see here, the statistical support was in favor of an increasing trend followed by a decreasing trend. And this technique, the segmented regression, allows you to compute also the bend point, which you see by these bars in these figures, and the width of the bar is indicating 95% confidence interval. So we see that in many cases we see a very clear bend point. You do see statistical, you do see that the decreasing trend followed by decreasing trend is statistically supported, but the position of the bend point is less supported. So we see this trend everywhere for plants and for the other organisms we have been looking at. So the first conclusion is that there is a universal non-monotonic relationship between compactness and expression level. And now, when we looked at compactness, we looked at coding parts at the length of exons, and we looked at non-coding parts, length of introns, and we wanted to see if they both increase and decrease in their length in a coordinated manner. So this is what you get if you just take the CDS length, the length of the coding part, and measure correlation with total intron length. So in all cases, you get positive, highly significant correlation. But then you can remeasure this correlation using the information about expression level categories. You measure the correlation in the between the different expression categories, and what you get is this. So the correlation is in human, in Drosophila, and in C. elegans is the increase in correlation is statistically significant. It is, you also see increase, although it is not significant for Arabidopsis. And what it tells you is that coding and non-coding parts of the gene increase and decrease their length in a coordinated fashion. So they increase together. So there is some coordination, but it is the way that they do it is not identical. First thing that you can look in this is the bend point, and you see that in some cases it is very different between the coding part and the non-coding. So this first indication that are similar in the general trend, but not all. And a second thing that you can look at is the mean exon length and mean intron length. And this is if you plot it. So you do see a very clear non-monotonic trend for introns, but for exons, you see actually a monotonic decrease or increase, and we will get to this point a little bit later. So there is some coordination in the way the coding and uncoding parts increase or decrease their length, but the forces do not seem to be identical. There seem to be differences in the forces that are acting on the coding and non-coding parts. In particular, introns tend to be elongated in a much more pronounced way for genes with very low expression. The next thing that we wanted to test on this data is which of the original explanations, the selection hypothesis versus the genomic design hypothesis, explain more this trend. So we wanted to measure expression breadth, so the number of tissues in which the gene is present. The usual way to do it is just to set a threshold and say that if the expression of the gene is more than a certain threshold, it is present in the tissue. In the threshold, it is absent. We try to do it in a more accurate way by doing the following procedure. Suppose this is our data matrix with, again, the rows are the genes and the columns are the different tissues or conditions. And what we do, we look at each gene and take its expression values, and we assume that each gene has two states. It's either present in the tissue or it is absent. And each of these two states is characterized by a typical probability distribution. So if the gene is absent, you would see some noise in the probability distribution of detecting expression is something that should be monotonically decreasing like this. And if it is present, you expect something more of a bell-shaped. And in order to model this, we use the mixture of gamma distributions, because gamma distribution can describe those two behaviors. So for each gene, we did a mixture of gamma procedure to estimate these two distributions. And then, after doing so, we can replace each number in this matrix, and and the number can indicate what is the probability that the gene is present in this tissue. So we replace the measured expression level by a number which has a probabilistic meaning, telling us what is the probability that the gene is present in this tissue. And then we redefine the expression breadth of a gene by just summing these probabilities. So this is what we took as expression breadth of a gene. And when you look at how the different compactness behave as a function of the expression breadth, this is what you see. This is human. And you see that you do see some non-monotonic trend, but it is way less pronounced than when you look at the data versus expression level. So our conclusion is that a major role of selection rather than genomic design in shaping gene architecture. And I also want to talk about intron density. So intron density means how many introns are per unit coding length. And if you plot intron density versus expression level theory, this is what you see. We will leave C. elegans aside for a minute because it is behaving in an opposite way to the other organism. But we see, maybe interestingly, that for all other organisms, intron density becomes higher with the expression level, meaning that highly expressed genes tend to have more introns per unit length than genes that are expressed in lower levels. And this can be re-verified if we a completely different data set where we took several thousand genes for which we measured different global system level parameters like global measures of activity in protein-protein interactions, in genetic interactions, and other variables, including some estimate for the average expression level of the genes. And for these variables that were measured in a different work on a different data set, we developed a more evolution, and from this model we could estimate for every gene the rates of intron gain and intron loss, and this is what you see here. So this is the intron gain rate, and this is the intron loss rate, and therefore for every gene we can correlate these intron gain and loss rate with the other variables. And what you see here in blue circles is those correlations are statistically significant. And what is interesting to this talk is to look at expression level, and what you see here is that genes that are highly expressed tend to have higher intron gain rates and lower intron loss rates. And again, this leads to the same conclusion that high expressed genes tend to accumulate introns. I believe this just that introns have important role in boosting expression levels. But the last thing I want to show here is a speculation of why this non-monotonic trend is happening. So what I think is that the decreasing trend is indeed due to selection, not necessarily selection for time and energy, which seem to be not such a huge burden on the cell, but maybe selection to get more accurate transcript splicing and other things. So it is selection to minimize error rate maybe, and the increasing part might be maybe it can be related to the previous talk. It is some kind of neofunctionalization, not by gene duplication, but maybe by gene elongation, where you if a gene is elongated for one reason or another, if something is dependent, it has a higher probability to interact maybe with other genes and to have higher expression levels. And thank you for listening. And this work was done in collaboration with Eugene Koonin from the NCBI.
You have to go to the microphone, I'm sorry.
For the accumulation of introns, do you think it might have something to do with program alternative splicing?
This particular accumulation of introns is happening in highly expressed genes, not necessarily related to alternative splicing. Of course, I think that accumulation of introns is beneficial also because of alternative splicing. But in this particular case, I think that it suggests that it helps in boosting up the expression levels. I wouldn't say necessarily because of alternative splicing. Maybe, but this data doesn't tell anything about this.
Several years ago, there was a paper stating that rapidly regulated genes are intron poor. So it's
what genes?
Rapidly regulated. So it's negative correlation between speed expression, expression gradient, and a number of introns per 1,000 nucleotides. And these guys, I think, achieved quite good correlations. So have you compared these two measures, not just expression, but the gradient of expression, and which one has explanation?
Yeah, that's a good point. Indeed, it was hypothesized that you need to change the expression level fast over time. You will have selection for short introns or for elimination of introns whatsoever. I didn't test this with respect to this data, but this is another thing that might be going on.
I think that based on the way that you sort of originally presented the motivation as being efficiency related to cost of adding nucleotides and things like that, I was sort of expecting one of the features that you were going to compare to be something like the efficiency or the compactness rather than the length of the protein or the length of the coding sequence, the length of the transcript. Because if you think of housekeeping genes or things like that, I mean, it seems like you're going to have proteins that are necessary to be certain lengths in order to do things, and that you can't necessarily compact out that sort of behavior. But what you could compact out, I mean, if you make the likely extremely false assumption that, you know, a lot of introns don't actually do things, then what you'd want to compare, a feature that you'd want to compare is something like the efficiency or the compactness of genes rather than the total length. So something like the length of the coding sequence is the length of the exons divided by the total length of the transcript. And I think that would be a really, really interesting feature to compare in the sort of analyses that you were doing. And then you could look for outliers in that have more intron density than more of their total intron. They were transcribing more than they were translating than you'd expect, and that might hint to regulatory sequences in those introns. Just something that would be interesting to look at.
Yeah, well, okay, what you say is relevant mostly for the exonic part, the coding part, and yeah, you could look, I guess, at the ratio between total exon length and total transcript length, and maybe that longer introns are less of a burden if the exons themselves are much longer. We didn't look at it. So I cannot tell you how the ratio behaves.
Yeah, I just think that might be a better representation of compactness than the things that you were measuring.
Well, yeah, but we thought that total length would be an ultimate measure of how much energy and time it takes you to transcribe these elements.
I guess what I was saying was that the length of exons might not be a good representation of selectable compactness, whereas introns you might expect to be more available to be shortened with evolutionary changes.
You actually see it in the data. The introns are much more, the trend is much more pronounced for introns than for exons, but you do see for exons too, yeah.
Yeah, you might expect the lengths to be much more dynamic, and rather than like the number of introns, the actual.
Can we have the last question?
Yeah. Thank you for this nice talk. So actually you used the different tissues and conditions to get a correlation of different intron exon length with this expression breadth, expression rate, etc. But I'm just curious to know if you have tried to see this or if you have found any interesting things for as the tissue specificity is concerned. So if you see in a tissue specific manner, do you see different natures of this overall trend, or you see the same nature even in each and every different tissues?
So we didn't look at tissue specific, we didn't look at each tissue separately, but the compactness is measured, of course, all over. So actually this is one of the things that we were thinking of doing next, is that, tissues differ in the total mass or the total number of cells in them. So expression in different tissues might give rise to different selective forces, like to the overall expression in the organism. So we want to look at it, but yeah.
Yeah, thank you. I'm just interested because you were saying finally the selection is driving this kind of overall trend. So it must be, there must be some tissue specificity or condition specificity there.
Okay, thanks again, Liran. Thank you for being here.